Hello everyone, and thank you for coming to my poster presentation. My research project deals with the effects of erythrocyte antigen density and dose on the development of IgM and IgG antibodies. So we all know that exposure to foreign antigens on red blood cells can lead to alloimmunization and the generation of alloantibodies. However, even though transfusion recipients are exposed to multiple foreign antigens, only about 2 to 6% of them become alloimmunized. Currently, we are unable to predict which patients will make alloantibodies and are therefore responders, and which patients will not. However, we do know that a variety of factors contribute to red blood cell alloimmunization, including factors such as antigen disparity between donor and recipient, underlying medical conditions, or prior transfusions. We also know that intrinsic red blood cell antigen factors can also contribute and play an important role in immunogenicity. And one of the best examples of a red blood cell antigen that's present at different densities is IHD. Exposure of an IHD negative mother to an IHD positive fetus red blood cell during pregnancy can lead to hemolytic disease of the fetus and newborn. So although we know that IHD is expressed at different levels and that this antigen density can contribute to immune responses, this has not been further thoroughly examined in the context of HDFN. To address this, I generated a novel mouse model, which allows me to alter the density of the model antigen HENNEC lysozyme, ultimately generating red blood cells that express low, medium, and high levels of HEL. By using these cells and injecting them into wild-type mice, I am able to examine the relationship between antigen density and immune responses. Each mice receives 10 to the 6, 10 to the 7, or 10 to the 8 HEL red blood cells at day 0, and then another one at day 28. Each week, their serum is analyzed via ELISA to detect IgM and IgG antibodies. So the coupling process of the HEL to the red blood cells generated red blood cells that express different quantities of HEL. So you can see there's a clear difference between the high, medium, and low cells here. This is an in vitro flow analysis of the cells that are used for injection. And this is supported by our antigen quantification analysis, which examined the antibody binding capacity of these cells. And you can see again that there's a clear difference in the expression levels. So now if we examine these cells when we inject them, all of these graphs are set up exactly the same way. You have the IgM response in blue on the left y-axis and the IgG response on the right y-axis. The blue dotted line is the mean and 2 standard deviations of the control mice, and these are mice that receive normal red blood cells without any antigen. And the red line is the same for the IgG response. So anything above is considered a productive immune response. And as you can see here in the top line, we have 10 to the 6 cells injected at low, medium, and HEL. There is no productive IgM or IgG responses being made. Even after the re-challenge, you only get a productive IgG response with the highest antigen density. If you inject 10 to the 7 cells here, you can see that you can get good IgM responses across all the different densities, but you only get IgG responses for the high HEL cells and the medium HEL cells after the secondary immunization. Lastly, if you inject 10 to the 8 cells, you get good IgM and IgG responses across the board, and you can also see that with increasing antigen density, the IgM response actually goes down. So now I also wanted to know whether the secondary immunization not only increases the IgG response, but also changes it. So I looked at the IgG subclass distribution as a percentage of the whole IgG response on day 7 and 35. And I found that on day 7, you get a contribution largely of IgG2B and 3, which are subclasses associated with a T-cell independent response. Whereas on day 35, you get a large contribution of IgG1 and 2C, which are subclasses associated with a T-cell dependent response. So overall, our results show that productive immune responses only occur above an antigen threshold, and this threshold is made up by antigen density and dose. So mice can either respond to a low density antigen delivered at high dosage or a high density antigen delivered at low dosage. And it interestingly also shows that weak primary immunization may not actually induce tolerance in mice with this antigen. And lastly, a re-challenge increased the IgG response and also altered the IgG subclass profile. Thank you very much for listening.